So, good morning. Today I'm going to talk about complex object and distribution of items, not statistical distributions in the sense of distribution functions, but more how objects are distributed. That should move forward. Yeah, now. So, some literature first. I think a lot of the material today can be actually found in Pierre Soil's book about morphological image processing. I see here I forgot to add the title, but it's on other slides as well, so you probably find it. John Ross also gives you some indication and morphometry in R. I had it a couple of times already. Also provides some information about these things. Some papers and sites to look at, a long list. But in principle, so here we have today's outline, what we want to do, and then the main topics that we are going to look at. Actually, in this list, the distribution is missing, which is actually the first topic. Then we look at skeletons and watershed segmentation, which is kind of the better one instead of component labeling. And yeah, that's what we're going to do today. So what we have done until now, we actually pretty far progressed. We have looked into what images are, how to collect the data sets, some filtering, how to segment and identify objects, and then finally also doing some shape analysis. And today we are going to different kind of objects. In the previous, when we did shape analysis, we identified single items that we could easily analyze, but now there are different approaches that we are going to look into. So we have, again, our test images, and we want to find some distribution of particles, for example. Here you see we could do the segmentation, we could look at distance maps, but they don't really tell how these things are distributed relative to each other, and also maybe see how the distribution change when the objects grow, etc. That are objects that or tasks that we haven't really been addressing yet. So looking at the distribution of objects, first of all, we have to define the right questions. And maybe we want to know how many cells are alive. And in this example, we have a living cell, which is nice, convex, but the dead cell is more spiky and fuzzy edges. And these are the ones we want to identify. And now it's a question how they are mixed. They could be mixed in clusters, or actually clustered that you have the healthy cells on one side and the dead cells on the other side. But it could also be that the dead cells are enclosed in clusters of healthy cells, or it could be a complete random mix between them. And this is what we want to identify. And one way is to look into how to identify. You have some cell cultures in a microscope, for example, dyeing culture. Then the cells, as you can already see by eye, they are more sparsely distributed. And in the active culture, then they are much more denser packed. And well, you can see it in two images by the eye, but if you start having series of many of these images, it's not so fun anymore to do the swiping, living or dying culture. So this is what we want to do. And the question is how we can compare these regions in the samples. And the solution is we want to get away from just counting cells. Okay, you can count cells, but if you want to do this characterization in your culture, you may do the subdividing it into regions, and you count the cells in each region. That is a way, of course, but that could be bit cumbersome and maybe not the idea. And also maybe we also want to avoid comparing orientations of the things, because it doesn't matter how the cells are oriented. So let's look into the different ways. We have some metrics. You can look at the nearest neighbor. So that requires that you actually have identified the cells, and then you can do some kind of neighborhood calculation. Would be, I would guess, an n² complexity of that. A small data set, so that's feasible. You can also look at point of center based tessellation, or how they are aligned. Voxel based, you can go for Voronoi tessellation, neighborhood counting, or some endpoint correlation function. So look at the nearest neighbor distribution. Here we have one culture. You see the dots are distributed in different ways, and if you look at the distance to the nearest neighbor, we get some distribution. I don't know, maybe it's Poisson distributed, potentially, I don't know. But it looks like it has more heavy starting mode, and then there is a tail going out in this direction. But for now, we have too little data for saying anything, and it's relatively sparsely distributed, so we should, in principle, have much more data to make a conclusion from this distribution. But at least it gives us something. A different way would be to look at the Voronoi tessellation, and that is based on the distance transforming principle. And what you do is you compute the distance transform from all these spots, and then on the ridges of the distance transform, you may remember that if you have a distance transform, there is always a maximum ridge between them, and that is where you put these lines. And now we have, instead of just distances, we have support areas. So the whole area where the distance is the maximum, is the shortest to this dot, is calculated for each object. And when we do that and look at the histogram, we get a much more unique distribution of areas than we got with the distances. So this may be a more robust thing to work with. Next lecture, I'm going to actually go into a little bit of the statistical analysis, comparing different cultures. But for today, we just stay with the tessellation. So what we can look at is, so we leave that now. And now we want to look at structures of different objects. And we already know we can compute the area. It's easy. Perimeter, a little bit more tricky. Orientation, position, and things like that. That was the previous lecture. And now we want to go into skeletonization and advanced object labeling. And first of all, skeletonization is something that is useful when we have some kind of network, very thin structures. And it could be street maps, rivers, routes, blood vessels, things like that, can be reduced into very nice skeletons. And the skeleton is, no, I'm telling that on a later slide. So what we want to know about the structures is how these are branched, how they are connected with each other, maybe if there are loops. So in a street map, you certainly have loops. In the river network, you may have loops because you have islands maybe in the river. In biological structures like roots and blood vessels, you shouldn't have any loops, then something is wrong. And we want to describe this in a more abstract, simplified manner. Maybe also quantify the tortuosity, which I'll come to later, and tell us something about how the structures are branching. Sorry about that. So the network, the skeleton is the minimal structure that describes the object. And if we take this horse, the skeleton of the horse in a image processing manner looks like this one, like kind of a line drawing by a child. But you can see actually it contains all the interesting features. It has 2 legs back, 2 legs in the front, has 2 ears, has a nose, and also a tail. And in the tail, it's a bit fuzzy because you can see here that there are some kind of gaps into the structure. But in principle, you could grow it out and get the horse back again. Probably a bit fatter horse, but anyway, you can grow it out to become a horse again. So this is the kind of minimal structure we are looking for. And the horse may not be the smartest object to do it on, but if we come back thinking on route networks or blood vessels, street maps, etc., then it actually makes sense to reduce it in this way. And the extreme case is a sphere, which reduces to a single point if it's a clean sphere. Otherwise, it may also end up in some kind of fuzzy structure. There are also very many different algorithms to do the skeletonization, and which one you choose depends on how you want to describe your structure. As I said, the sphere could reduce to a single dot, but it could also reduce into something that always connects to the surface with that requirement, and then it maybe would end up in some kind of cross or snowflake structure. So it depends a little bit on what you require from your skeleton. So let's look on some real images to see how the skeletonization process works, and one algorithm, a couple of algorithms how to do it. So let's take this aerial image of a street neighborhood. It's actually labeled, so that's great. Here we see what is the expected street map, and now we'll see how well we can reduce it into a skeleton. The first step is this is a bit too much, will be too cluttered, so we reduce it a bit, and we just take this main street with 2 branches, which we zoom in, and here are the ground truth roads as marked up. And this is what we are going to work with. So let's find the roads. First of all, this is a color image, and we haven't looked much into segmenting color images yet, and we're not going to do much about it anyway. But anyway, in color images, we have 3 channels: red, green, and blue. And they compose all the colors. There are also other color spaces that out there, but this is the one that is usually used when you save images as files. So let's do the pair plot. We have the red, green, and blue channel histograms here, and here we can see how they are connected. I don't really see, so there is something very bright coming out here in the blue channel, but not, and also in the green channel actually. But that's not enough really to tell. So let's do something else. We switch to a different color space, saturation and value, which is a different way of representing colors. In principle, you set the spectrum is an angle, and you get your colors as an angle, either in 360 degrees, 2 pi, or even normalize 0 to 1. And the saturation is how strong the color is, and the hue is how dark it is. No, wait. I'm probably mixing it up now. Yeah, anyway. What we are going to look at is the value, also the V value. So we do the conversion into different color space, do a segmentation on that one. We can go back and see. So we set the threshold around 0.4 around here. So we have some kind of bump, which is a small representation in the image towards the brighter values. And if we even go back to, hello, here, the street, the gray is somewhat brighter than the green areas. So let's make this assumption. So, and setting this threshold, it's just a hard threshold without any smart algorithms. Then we get this kind of segmentation. Yeah, the street comes out, but we also get some more clutter. In this case, it's the rooftops of the houses mainly, and well, here's some kind of fuzziness from probably from the scanning. I don't know, which also comes out as street material. So this is too much, so we need to reduce it. And the first step is just to identify the street itself. Here we make the assumption that the street is the largest structure, so we just do the component labeling as we know from last lecture. We see here the labels of the different structures, and we see that structure 1 is the one that we want. These two ones, they look like they are disconnected, but if you look at the full image, you can see that there is a connection between them. So this is just downsampled to see the values of the labels, because you can't see it in this one. That would be too cluttered. So let's do some cleaning. We know already that we can do cleaning with the help of morphological opening and closing. And here I used different sizes of the footprint. So I have disk 1 and disk 2.5, because I want to clean different sizes of structures in this street. With the cleaning, I get rid of some connections. You can see here now that after the closing and opening, we can remove at least this blob and this blob. So if we run the labeling, we have a more reduced street map than before, which was, and also with less clutter. You can see in this region over here, there are very small, tiny little peaks. And these peaks, they are really annoying when you start computing the distance map and also the skeleton. The skeleton can be really wild and unpruned if you have too many of these tiny details. So, the first take of a skeletonization, we want to have the middle line. And one operation that we have done already, which produces this, is the distance transform. So if we compute the distance map of this structure, we can see that we have some kind of middle line that could be used, but still it's not a skeleton, it's still some kind of gray level image. So we need to do something more. And that thing would be, for example, computing the Laplacian of the image, and the kernel would look like this. And if we run that one, we can see very nicely that it captures the middle line. It also captures the edges, which we are not really interested in. And you can also see that there are a lot of tiny branches going out everywhere. And these tiny branches, they are caused by all this little ripple on the surface of this street bitmap. So, to create the skeleton, we have to do some pruning, and we have to threshold the Laplacian of the distance map. And in principle, we can have two criteria. One is the minimum distance. it cannot be a branch if it's too close to the surface. And the other one is that the minimum slope should be also defined. And when we do this, we get these two criteria, and this is again also just empirical values set, but looking at the images, these are the pixels that would be considered as the skeleton. So if we look at what happens, is first we have the distance map, the Laplacian of it, and then by this filtering we get, or thresholding, we get this map, which is kind of a skeleton, but you can see there are very many branches, and that is the tricky thing when you start doing skeletonization. You get too many branches, just a little disturbing pixel on the surface, or a misclassified inside produces loops and a lot of tiny branches around. So this is what we want to avoid. And there are, as I said before, also very many different algorithms. So one is called morphological thinning, and that is essentially that you apply an erosion until you reach the minimum thickness. So you erode and erode, and if a pixel finally is the minimal, then you stop eroding, and then you can iterate that as many times as needed. And in a scikit image, you just can say max iteration as a parameter. If you set infinity, it really reduces down to a skeleton. There are also other algorithms, but anyway. So here you can see the original, what we went from. This is a single iteration of the morphological thinning. Then we run yet an opening on that one. And the reason for that is we want to get rid of all these tiny things, which we had before. And that looks already a lot cleaner. And the little bit of problem is we have two misclassified pixels inside here, and they really come out as two loops in the image. But anyway, this skeleton looks a lot better than the one we had here. So it's already an improvement. So let's run on and see what we can do with this. So now let's say we are happy with the skeleton as we got it from the previous operation. Now we want to know something about the skeleton. Until now we just have some pixels corresponding to the middle line. But now we want to know if there are what each pixel on the skeleton, what it actually mean. And a way to do that is to apply a convolution to the skeleton. And the convolution with the box filter essentially is just the sum of the pixels in its neighborhood. And the interesting thing is that the sum tells already something about the neighborhood. Here I just did the convolution, and you got a wider skeleton. We don't want a wide one, so what we do is just mask the central line, the skeleton, and then we get this junction count skeleton. And well, the resolution and light maybe doesn't really tell you, but you have different colors in here. You have blue out on the tail points, and there should be some gray also where we have branching, and this is the information we want to extract. And let's look at what it does to us. So zoom in, and then you can really see it. So here you have an endpoint, got the label 2. You have some junction points, they got label 4. you have just line segments, they get the label 3. So now you can identify that you have line segments, you have connection points, and you have endpoints. And this is the information we want to know about the skeleton. And now looking at it, we can see that 0 means the background, 1 is an isolated point, so it's just an island. 2 is an endpoint, then we have the line segment, 3-way junction, and 4-way junction. So these are the types that we can identify with that kind of filter kernel. You can even make it more complicated if you want to know if the branch is pointing up or down or sideways or something. And then you can do different kind of kernel. So this would be a 4-connected neighborhood, and this would be an 8-connected neighborhood. And the reason why I'm doing this is that every pixel, in principle, gets a binary map, which is where each bit corresponds to a pixel in the neighborhood. And then you can do some kind of either you can actually do bit sequence analysis and see if it's the how the branching looks like, or you can just use it with a lookup table. Of course, this looks much more detailed. The colors are not sufficient to really tell the difference, but you can see that we have horizontal pixels or diagonal pixels, they get a 16, whereas the vertical pixels, they get a 21, and so on. So you can really tell the local orientation with this kind of mapping, which you couldn't do with the help of the first one. Which you use, again, is up to what you need. So that you have to decide yourself, but this is a way of doing the analysis. So we had already a rather nice skeleton, but you can even do further pruning to make it even cleaner, because you had some small branches which are not really good. So what we could do is maybe looking into branch length or branch width and use that to tell how to prune the skeleton. And looking first at the length of a segment, that is pretty easy, because you can just label all the items that we got. First, actually, we remove all the connecting points, so that would be the number 3. No, we select only the number 3, which are the line segments. And then we can identify or compute the area of each, which is actually the length. And then you can see here the distribution of length along for the different labels. And here the skeleton is labeled with its line length. And you can see some are very long, others, the dark ones, they are the short ones. So it's a way to label it. This is written out in code. You could also use the region properties to compute the area of these, if you would like to. So let's look at the pruned skeleton. We can see that if the label length is greater than 5 and the object is let's see, these are the junctions, and then we have the line element. But so we can prune it even more. So we took out now all the pieces that were too short, and you can see some endpoints still in this image. So those we also want to get rid of. But here you can see what the pruned skeleton looks like versus the ground truth. Almost getting there. What we managed to do was to remove at least one loop. There is still one loop left, but yeah, that one I think is going to be very stubborn. We can also look at the segment width using that, because if it's too narrow, it's not likely to be a street. So that could also be a way to put the analysis on the pruning. And here we have the structure width, and that starts looking really good. We even got rid of the loop. So this is getting better. And if we compare this one to the original medial axis, we are really close to what we want to have. So this is good. There is, of course, always different ways of doing the analysis. The example I did now was mainly to show all the pruning effects, but you can already get it much cleaner by doing some other choices. So one is changing the segmentation method. We only used one color channel, and we can also use some other morphological filters to do the cleaning. And the first thing is to try using the k-means algorithm and using all three color channels. And with that, we see here we have the k-means labels, and we extract label 0, which is the street. And when we extract the street from that one and do some closing and opening, you see already that this is a much nicer segmentation. And that brings us certainly to the conclusion that think twice in how you do the segmentation. Here we can see now from the basic segmentation we got that with a lot of clutter, and only using this k-means based on the color space, we get a much cleaner street. We could, of course, continue doing all the pruning that we did before, if we would like to. Now, we got a nice street, and now we want to find some topology, seeing how things are hanging together, because right now we have a bag of endpoints, we have a bag of line segments, we have a bag of junctions, but they don't mean anything. They're just pieces. So now we want to get some idea on how to do the topology. And here we have those pieces of the street network marked up. And now we want to have an algorithm to find how to connect the pieces. So let's say we have an image, a very small image, 3 by 4, where we have nodes. These are the ends and edges. And the principle is that first we label the nodes and edges separately. So we have node 1 and 2. We have here edge 1 and 2. And now we want to know how the different edges are connected with the node. And if we apply an dilation operation on the edges, we will see an overlap, which is useful for us. And what we want to have is this connection, and here we have the nodes, and this is the line that we want to make a connection. So let's do the dilation. We have the node images, we have them labeled, and if we grow the first node, it looks like this. And then we check which of the edges are actually overlapping with that one, and that we do, again, same routine. We have the edges, we label them, and we see that this block corresponds to the first label. And there is a clear overlap between those. So we make the connection saying that node 1 is connected with edge 1. If we go on, we would do the same thing from with the node 2 and edge 2, and then we will see how the things are connected. And with that, we can do some coding. We have first all our edges and nodes, which are put into to a dictionary where I have the positions and the width of each element, each node. And then there is a lot more code that in the end produces a dictionary with start and end points. We have the length, we have the Euclidean distance, and we have the max width, mean width, and so on. That's all in a table that we can look at like this. This one is still not connected, but we have added a bit more information about the edges. And when we put this information together using this overlapping algorithm, we can now make a connectivity matrix where we see how node A and B are connected with each other. And the central line is just the same node. And here you can see that node 2 is connected with node 3, and so on. So you can see how the things are connected, and it seems like in most cases everything is very narrow connected, but there are some places where you have it disconnected, and node 5, I believe, is one of these side streets, which then is disconnected in the labeling sequence. But typically you should have very narrow connectivity map between the different segments or the different nodes. So now that you have got this, we can start doing measurements on the skeleton. We have one quantity called tortuosity, which is the ratio between the curve length, however that would be, and the straight Euclidean distance between the two endpoints of that segment. And if you have low tortuosity, it's just a straight line. Goes from then you have the same curve length as the Euclidean distance, and then you would have a tortuosity very close to 1. If you have high tortuosity, then it's more curvy. Then the curve distance is maybe several times longer than the Euclidean distance from point to point. And that can tell you something about what your network is looking like. So that could be relevant for looking at blood vessel analysis, route analysis, also maybe seeing if you have meandering or straight rivers or channels, canals like this. So there are different information you can extract from this. Looking at the tortuosity of the street, most pieces, they have essentially 1, a little bit more. They are not perfectly straight. Now my red dots are not very visible, but you can look at your notes and see that I plotted some red dots on top of the blue segments. You can see that there is a little wiggling around there. And in some cases, apparently it makes a loop. I think it's close to this region. There is a lot of looping happening, and also here. And they get then higher tortuosity. Then you can also visualize the network in different ways. When you have a very long, straight network, it's maybe not so easy to visualize it. You would need 2 pages to show it. Then you can use this kind of randomly organized graph, which preserves the branching topology and also length, but then it curves in a way that it actually fits in the figure. So that could be a different way of doing it. Then also, when you want to visualize it, you could also add more information to each line segment. You could have the color opacity or something, saying the thickness. You can say the length by another color, connectivity by a third color, etc. So, or different sizes of glyphs. That is up to your creativity. And again, the purpose of your visualization, what makes sense to do. So we can, of course, also looking at the topology in the analysis. Here we can see how many nodes are connected with how many edges. And most nodes are connected to, actually it should be +1, I think. Or no, these are the endpoints. So all the endpoints only have one node. Then we have small line segments, we have more junctions, and so on. So this counts, and so you can see how is the distribution, and that can also tell you maybe about something about the health of a system, that if it branches a lot, then there is a certain condition. If it branches little, then there's another condition. So that is how you could use that. And of course, now I just showed the skeletons in 2D. It's easiest to show, but they of course also work in 3D. And again, the requirement is that the object topology must be preserved. And when you have 3D, you have a cube around each voxel, so the neighborhood is more difficult to analyze, and that also has an impact on which algorithm or how you define the skeleton. There is a nice overview of skeletonization algorithms here. And in some cases, there are skeletons that also allow plates or planes connecting the different branching points. So the branch points are expanded into planes. Depends again on what you want to do or the definition of the metrics, how you define it. Oops. Oh, I went to that one. That was a feature, okay. Just a moment. No. So, that wasn't meant to happen. So let's go on. So here is a 3D skeleton of a root network. And why we want to look at a network of roots is, for example, looking at soil stability, water uptake regions, and maybe also how the root networks is growing under different conditions. And what we do when we analyze the network is, of course, the segment length, how much it branches, also branch orientation. Some root networks, they grow pretty laterally and do the branching in that way. Others go downwards. And of course also the tortuosity. Another thing would be to look at the influence volume. So that's in principle a convex hull of the whole root network or fragments of the root network. And this one would actually be a little movie. So here we can see this little root network. And I have a data set here, so we can load it. The root network is already segmented. And if we look at single slices, it doesn't really give us much information. You just see some little dots in different slices because the roots are thin structures, so we can't really tell much. So what we do instead is looking at the projection in different directions. And then you can see a 2D projection. So here's the XY looking from the top. And then you have the side view, different side views, and there you can also see the how the root network looks like. This we can skeletonize with the help of skeletonize function. And yeah, then you get the skeletonized network. Again, here is the max projection to see what it looks like. Here I use the max projection because it's only single black and white pixels, and they would actually just get under in the amount of black pixels. So that's why I use the max. Yeah, and here we see a little bit a root skeleton from this region here. And well, that's a good timing for making a break.